Good morning. I apologize for coming late, and let me start with the talk. So, I've been working in eye genetics for quite some time, for almost 2.5 decades. And let me tell you what is happening currently with all this in India. My talk will be structured as follows: that I'll talk about the basics of ophthalmic genetics first, and then I'll talk about how we apply that in genetic counseling, and why we have to do research in ophthalmic genetics, and then what is the translation that is happening in the field, and also that what we are currently doing, about which Dr. Tandon gave a brief description now. So, if you take ophthalmic genetic disorders, you can classify them broadly into 4 different categories of chromosomal disorders, single gene disorders, complex disorders, and there's one more category called mitochondrial disorders. But they are very rare because you can't survive with mitochondrial mutation, and therefore most of the fetus are aborted, and that's what the thinking is. But complex disorders are much more common in any population that you might take. So from the lid onwards till the optic nerve, there are many disorders that affect the eye, and it can be classified as Mendelian or complex disorders and mitochondrial disorders. Now, if you try to take a list of these disorders, there are about nearly 500 plus genes so far identified, which directly causes a particular sign and symptom and a disease in a patient as far as ophthalmology is concerned. So you can obviously see that cornea and retina takes quite a large portion of these genes, for the simple reason that the anatomy, physiology and biochemistry of these two structures in the eye is one of the most complicated. Now I'm going to focus only on retina because that's where I've worked mostly. And if you take the retina and the RPE, which are two structures that you cannot separate each other from, you can see that there are distinctive functions of the retinal pigment epithelium as well as the neuroretina. So neuroretina is basically a neurovascular unit. So the vascular unit has its own problems like diabetic retinopathy and so on, which are also genetically influenced. But I'm going to stick to the neuroretina today and then the retinal pigment epithelium. So when light strikes the rhodopsin, so therefore in neuroretina and the phototransduction pathway, there are only 4 key players, and rest of them are adjuvant players in the entire phototransduction pathway. So it starts off with rhodopsin and then moves on to transducin, which again stimulates the phosphodiesterase, and ultimately that opens the or closes the cyclic GMP gated channel. Several genes are involved in this pathway, and in this pathway what happens is that the 11-cis retinol is actually converted to all-trans retinol at the end of this phototransduction pathway, and that has to be recycled, and therefore the all-trans retinol is recycled back into 11-cis retinol in the vitamin A recycling process in the retinal pigment epithelium. But besides that, there are a lot of scavenging function that the retinal pigment epithelium does, and therefore you have a bunch of genes working in the retinal pigment epithelium as well as in the neuroretina, which if they don't function properly, if they have a mutation, that leads on to various retinal degenerative diseases. So as I told you, there are 200 plus genes so far identified which causes one form of retinal degeneration or the other. But the complexity of all this starts when you try to interface yourself with your patient and try to do genetic counseling. So as you can see here, a lot of these genes overlap with the phenotype. So therefore you have a huge amount of genetic as well as clinical heterogeneity in the entire spectrum of retinal degenerative diseases. So if you take the say LCA5 gene, so that causes Leber congenital amaurosis, and it can also cause cone rod dystrophies. Okay. Now this is something which has not been very clearly understood, that why isn't there any kind of a clear relationship between the genotype and the phenotype. But nevertheless, there are a lot of things that you can do in terms of counseling your patients when they come to you with the help of the ophthalmologist. So therefore, let me move from the first topic of basics to the genetic counseling, how we use this in an applied sense. So I do genetic counseling in different parts of India, at least primarily in Mumbai and Bangalore right now, but I've been doing quite a lot of genetic counseling in Chennai at Shankar Nethralaya for a long time. The field has moved completely from very basic genetic diagnostics to very complex diagnostics. It's a very, very powerful tool. If you look at the history of and the evolution of genetic testing with time, it all started with sickle cell disease. As you can see, that was in almost 1949. And we moved a long path in trying to identify a variety of genetic disorders in non-ophthalmological disorders. And currently we are sort of parked here where we can do a very powerful gene screening. So therefore, about 10 years ago in India, we could do one single gene at a time. And therefore, it is very laborious, time consuming. Today, you can actually screen the entire gene panel or the entire genome of a human being in less than a month for diagnostics. So therefore, the problem there is that you would obviously find at least about 6% of the genes showing some change, even in somebody who's quite normal. So that's where the problem is. So how do you interpret all this data is also evolving at this point of time. Now you don't, in a clinical situation, you don't really need to look at the entire genome of a human being to understand the genetic nature of the disease, as well as in genetic counseling them. Even if you take the whole genome of a human being, what is expressed is equivalent to a thin slice of an apple. Okay, so it's only about 1 to 2% of our entire genome is expressed into a protein, which ultimately has some physiological and biochemical function in different tissues of the body and produces a phenotype. So why we have only 1 to 2% expressing in a human being is not very clear, whereas if you take a bacteria, they're much more efficient. 100% of the genome is expressed. So nevertheless, in a clinical situation, you don't even need to take the thin slice of an apple, which means you look at about 22,000 genes that are there in human genome. But you can actually look at what is called as the clinical exome, which is about 7 to 8,000 genes. So the reason you look at only about 7 to 8,000 genes is that they produce some kind of a clinical phenotype. Okay, so it produces some disease. So if you do 10 genes by Sanger sequencing, or 15 genes or 20 genes, and then you do about 7,000 or 8,000 genes by next-gen sequencing, the cost will be equivalent, but you will gain a lot of time. Okay, so doing 20 genes, laborious, time consuming in a small laboratory in a clinical situation. Whereas if you outsource these things to laboratories which are well equipped for these technologies, then it's much easier for you to get the result and interpret. Once the clinician makes a diagnosis and then knows that this is a particular clinical condition, and then gives it a diagnostic label, then they take the sample of the patient to do next-gen sequencing. So there's a huge pipeline which is done, and usually it's better to outsource it because they are quite complex with multiple steps, about 100 steps or so in the laboratory. So once the data comes, about 4 to 8 GB data is generated in this process, which is huge amount of information. And then it is sort of analyzed through bioinformatics. So that's another pipeline. So you need a good set of people who are quite experienced in this analysis. And this kind of analysis is done real time with various public and private websites which maintains a catalog of diseases and mutations. So once that is done, you get a result. And I'll tell you 2 examples of me interacting with patients. These are expensive. Test in Indian terms, it can cost you anywhere from 30 to 35,000 rupees for a patient, which is about something like about $500 US. Now, once this is a patient who had retinoblastoma, so the mother had retinoblastoma, and one of her son had retinoblastoma. So the other 2 are actually, one is the father and the other one is the second child they have. So the mother had a heterozygous deletion of the RB1 gene, and she gave that chromosome to one of her son who also had retinoblastoma. She had one eye enucleated, whereas this boy was diagnosed quite early because there was an awareness in the family. So the reason I'm showing you this slide here is that this father wanted to have another child without retinoblastoma. Okay, so that's the reason he came to us, and we are currently handling him. The current pregnancy of this lady went into abortion, and so we still haven't done any prenatal diagnostics in this family. Now this is another patient who had retinitis pigmentosa. So I'm showing you this for the simple reason that how complex an analysis can be. So when this was screened, you could see that the patient had multiple mutations. Okay, so this was in GUSI2D gene. It was a homozygous mutation. In EYS, there was a heterozygous mutation which has been reported, and there's another mutation which is not reported, okay, with a particular clinical phenotype which was identified in this patient, and there was another one gene which also was heterozygous mutation change. So why I'm showing you this is that this was a consanguineous family, and therefore it was much easier for us to pick that the mutation has to be homozygous. It was an isolated patient, and therefore in scenarios like South India where you have consanguineous marriages, pedigrees are very important when you try to look at it, not only the clinical information. Now therefore this whole field of next-gen sequencing is sort of revolutionizing the whole field of Mendelian disorders through whole exome or clinical exome sequencing, and then sort of giving a much appropriate counseling to your patients. Now let me move on to the third segment, that why do you have to do research in an ophthalmic institute? So what's the benefit that you get? So let me give you an example in this. So this is actually a family which was reported by Ajoy Vincent at Narayanetralia. So it was a family with foveal hyperplasia. This was published in Eye, and then I literally entered Narayanetralia at this point of time, and then read this article, and then discussed with the clinician about this, and this is the clinical features. So I am not an ophthalmologist, so most of you can read this much better than me. But when we looked at the pedigree, this is the pedigree. Okay, it's a sort of a deceptive pedigree for somebody from a Western country, because obviously looking at this pedigree, of course these hatched ones are some other ocular problem, so which we can ignore at this point of time. And you can see that the father and 2 daughters are affected, whereas 1 daughter is normal, and you can see a consanguinity. And obviously it can be misinterpreted as autosomal dominant, but this is actually what is called a pseudo-dominant, where a recessive person gets married to a carrier, and then you have more of the children affected. Okay. So we do see quite a lot of these ones, and we took them for a study and gene mapped it, and then found a mutation in a gene called as SLC38A8. Okay, so this is our mutation, and at the same point of time, Chris Inglehorn in University of Leeds, so he was working on the same area, and then we had a discussion. We had a linkage at that point of time, not the mutation. So both the families had lot of, not both the families, he had multiple families from Europe, and those families and the family in Bangalore had similar phenotype and a location as well. So therefore we quickly screened the gene that they were suspecting, and we found this mutation, and these are the other mutations found. So this is actually a glutamine transporter, which is very important for neurotransmission in the photoreceptors. So therefore that's the knowledge that was gained by studying this family. So this is the publication that came out in the American Journal of Human Genetics. Of course, they did a lot of other functional studies. We didn't have the capability to do it. So they had morpholinos that they produced with zebrafish and showed some sort of clinical feature similar to humans. So therefore, all this research actually takes you from discovery to diagnostics and management. Okay, that's the pathway that ultimately the whole thing takes you. So, of course, I don't need to tell the audience here what this is. So that's the retina, and then the RP, as you can see, with the photoreceptors. So this slide is something I showed you earlier with the retinal pigment epithelium and the neuroretinal function. As I told you, there are multiple genes which are involved in this entire process, which we perceive as vision. And if you could see the function of a rod in dark and light, you can see that the entire phototransduction pathway regulates the opening and closing of this cyclic GMP gated channel. So in a normal situation, when you close your eye, the rod photoreceptor is depolarized, and once the light strikes the rhodopsin molecule, there's a pathway which I explained earlier, where it takes you from rhodopsin to transducin to phosphodiesterase to the cyclic GMP gated channel. So at which point of time this closes, okay, and prevents positive ions to get inside, at which point of time the rod is actually hyperpolarized, and therefore it passes the signal to the optic nerve. So it is at this point that SLC38A8 is involved in neurotransmission, which is something which was not understood earlier, and therefore was a discovery. So therefore, in the outer rim of the rod photoreceptor, and probably also in the cone, there are multiple proteins. As I told you, the key players are rhodopsin, which is sensitive to light, and transducin, which has components of alpha, beta, and gamma, and then phosphodiesterase, which has alpha, beta, gamma, and then that's the cyclic GMP gated channel. So each of this component of the protein is produced by a gene. Okay, so if you have a phosphodiesterase A mutation, then the process will stop here and it wouldn't go further. So therefore what happens is this gets into a vicious cycle, okay, since it doesn't move beyond this step. And therefore what happens is the rod gets confused, and therefore it sort of starts to destroy itself because of that dysfunction, and leads on to apoptosis and retinal degeneration. So that's what happens to all these mutations, which ultimately leads on to retinal degeneration. So where is this all leading to? Well, it's all leading to gene therapy and stem cell therapy, where they are trying to see that if we can actually help our patients by treating. So that's actually the global clinical trials going on. You can see that it's only about 1.4% of gene therapy trials which are actually ocular in nature. Of course, mostly adeno-associated virus is used for this process. So I'm coming to the last segment. I have 5 minutes. Okay. So in all this knowledge and initiative experience that we have gained, so what is that we are doing currently? So this is what Dr. Tandon explained right at the beginning, that we have formed iGenetics Consortium. So this was an initiative actually started by Dr. Radhika Tandon and Dr. Namrata Sharma, and then they pursued it and spoke to me, and we have really moved it much forwards. So in this small segment of my talk, what I'm going to tell is that we have started with 2 projects. One is on retina, another one is in cornea. So the cornea one is going to be managed by Dr. Radhika Tandon and her team. In the retina, we started off with studying retinitis pigmentosa. So this is something as a project, as you can see here. I'll tell you what this project is. There are 7 institutions which have joined together, and we are trying to do a project collectively, and we spoke with the Department of Biotechnology, Government of India, which is interested in looking at this for funding. So the 7 institutions are from Vanita Kumar from Guru Nanak Dev University, and then Dr. Manisha Agarwal from Shroff Sai Hospital, Dr. Natarajan from Aditya Jyot Eye Hospital, Mumbai, and Dr. Chitra Kandabiran from Hyderabad LV Prasad Eye Institute, Dr. Arka Ghosh from Narayan Nethralaya, Bangalore, and of course this is another unit of LV Prasad at Visakhapatnam. Then Dr. Sundaresan from Madurai Aravind, and Dr. Sri Priya and Saumitra from Shankar Nethralaya. So Shankar Nethralaya, LV Prasad also has a branch in Bhubaneswar, and Shankar Nethralaya and Aravind have branches in Kolkata, in this city. So they're running clinics here. So with these 7 institutions, we collectively are looking at about 70 to 80 lakh people in a year, which means about close to about a crore people spread all over India. And the team consists of 14 experts, both clinicians as well as scientists in various fields of in laboratory science. And collectively, 14 of us have about nearly 400 publications in vision sciences. So therefore, that's the title of the project that has been already submitted to the Department of Biotechnology. We are also going to look at a particular factor, because the prevalence of RP is 1 in 4,000, whereas in a South Indian population study by Dr. Sen et al. Shows that it's almost 1 in 1,000, and if you go to rural part of southern India, it's about 1 in 400 or less. The reason this could be a biased study, because this was not primarily oriented towards studying retinal degenerations, but it was actually a glaucoma study where there was an incidental finding. Therefore, the study could be biased, but not this much in difference that we think. We are not looking at the prevalence, but we are going to look at the various mutations and genes that are prevalent in India. Of course, that's the retinitis pigmentosa. So the study has 2 segments. One is to gene discovery of new genes in Indian population, and then mutations that are there. We are going to look at about 150 families. Still, this is not funded. We have applied for funding. And the second part is looking at the functional analysis and therapy. So going to create patient-derived iPSC cells And then we are going to look at the possibility of intervention with gene therapy. So that's the whole summary of my today's talk. So I started off with the basics, then told that how we apply it in genetic counseling, and then how this research and discovery leads on to diagnostics as well as probably therapy in retinal degenerations, and also a current study that we are trying to do with and applied for funding with Government of India. So I have to thank my group in Bangalore, which is under Dr. Natarajan, and of course Dr. Radhika Tandon is here. So that's the whole group where we would be collecting families with retinal degenerations for this particular study. And this is our Bangalore group. That's a set of clinicians, and this is headed by Dr. Bhujang Shakti, and then deputed by Dr. Rohit Shetty. And this is our research group, lab research group, and this is Dr. Arka Ghosh, who will do the gene therapy in this particular study, and that's Dr. Debashish Das, who's a stem cell expert. Thank you.